Hello students, I am Sobia Zafar from Hamdard University. In this module, we are going to discuss about the ex vivo testing used for the pharmaceuticals. The learning objectives of this module are: first, what is ex vivo testing? What are the different models used in ex vivo testing in oral and transdermal delivery? What are the different techniques for the ex vivo testing of products to be administered by ocular, buccal and nasal route? So before starting with the different techniques used in the ex vivo testing of pharmaceuticals, we shall discuss what is actually meant by ex vivo testing. In 1959, Russell and Birch performed a study based upon the philosophical concept of humanity, where they observed that some biological experiments could be classed as inhumane depending upon the levels of pain, distress and lasting harm experienced by the test animals. Their research provided the systematic basis of 3 R's, that is, replace, reduce and refine, the use of sentient beings in the experimental biology. This led to a journal expansion of the funding sources for ex vivo and in vitro alternative methods, reducing the dependency on live animal testing whilst also creating a political climate whereby the alternative procedures were incorporated into the federal and government legislation. In the ex vivo models, the experiments are carried out with the living functional tissues or organs isolated from an organism and cultivated in the artificial environment outside the organism under highly controlled conditions. These models mimic the different biological barriers that a therapeutic molecule has to overcome to finally reach its target organ or cell or receptor. A primary advantage is the ability to perform tests or measurements that are otherwise not possible or ethical in the living subjects. They not only characterize the permeability behavior of drug molecules in epithelial and endothelial tissues, but also helps in studying the drug delivery systems for improved delivery and enhanced absorption. The basic difference between the term in vitro and the ex vivo is that for ex vivo, the samples to be tested are extracted from the organism, while in vitro means the sample to be tested are obtained from a repository. The ex vivo studies involves the use of tissues, whereas the in vitro study involves the use of artificial media which resembles the biological fluids. Compared to the complex in vivo situation, the ex vivo models offer a fast, convenient approach with cost advantages. Most importantly, they can be standardized and automatized to be applicable to the high throughput screening. The different types of ex vivo models used in different routes of drug administration are discussed in the subsequent section in this module. The different types of ex vivo models used in different routes of drug administration are discussed in the subsequent section in this module. So the first part is the ex vivo studies in oral drug delivery. Oral route is the most preferred route of drug administration. The oral route, though associated with the greatest degree of patient compliance, as it ensures convenience, enables self-administration and offers great flexibility in dosage regimen. However, achieving effective drug delivery and minimizing off-target side effects is often challenging. One of the most important concerns of pharmaceutical industry while developing drugs for oral delivery is to have a good predictive model to assess gastrointestinal permeability in humans. Various systems have been developed varying to the degree mimicking in vivo conditions. Tissue-based models are an attractive tool to study the intestinal drug absorption. The possibility of using human intestinal tissues make these techniques even more attractive. The various ex vivo models used are discussed below. Ex vivo studies in horizontal diffusion chambers. The drug permeability studies across the ex vivo intestinal tissues are generally performed in diffusion chambers. Depending on the solution flux direction, diffusion chambers can be classified as horizontal, also known as side-by-side or async chambers, or vertical, called as France cell. The async chamber technique was initially developed to study transepithelial transport across the frog skin, but now widely used to evaluate intestinal drug permeability. They can vary in size, volume and area of exposed tissue. In this model, a section of intestinal tissue is excised and cut into small segments and opened to form flat epithelial sheet applied between the two halves of the chamber, which are filled with physiological buffer with continuous supply of carbogen, which is consisting of 95% oxygen and 5% carbon dioxide, and temperature control to 37 degrees centigrade. The carbogen ensures continuous adequate oxygenation of the tissue and creates fluid circulation in both compartments, reducing effects of unstirred water layer that jeopardizes solubility of hydrophobic compound. The advantages of such technique include it mimics the in vivo situation, allows study of regional differences in intestinal absorption, study of transepithelial drug transport with intestinal metabolism in the same model, requirement of low amounts of test compounds, conditions to which tissue is exposed is easily manipulated, presence of mucus layer in apical sites, allows to assess role of drug transporters in intestinal absorption. The possible disadvantages are dissection of epithelial tissue is difficult to perform and serosal muscle layers can only be partially removed. It is a time consuming process. The changes in morphology and functionality of the transporter proteins during removal and mounting of the tissues can also be observed. Coco et al. Used the horizontal diffusion chambers for the evaluation of uptake of polymeric nanoparticles containing ovalbumin as the model drug for the local delivery of molecules in the treatment of inflammatory bowel disease. Acute colitis was first introduced in female mice using dextran sodium sulfate. Mice were sacrificed at day 12 by cervical dislocation. After that, a mid-ventral abdominal incision, the colon was removed and immediately immersed in freshly prepared oxygenated Krebs-Ringer solution at room temperature. Colon was opened longitudinally along the mesenteric border and rinsed with Kr solution to remove the intestinal contents. The sample of colon was mounted between the two tapered fitted halves of the chamber, which comprised an Eppendorf tube and its cap with an internal window area of 0.35 cm square. The internal mucosal side volume contained 0.2 ml in the inner chamber of the Eppendorf tube, and the receiver compartment was filled with Kr solution and gassed with carbogen during the whole procedure. Furthermore, no gas inlet was provided in the apical side so as to mimic the low oxygen environment in the luminal side of the colon. The apical side of the chamber was initially filled with Kr solution at 37 degrees centigrade for 30 minutes and then cleaned and filled with test solution. 1 hour post application, the apical side of the chamber was washed with Kr solution and the colonic samples were removed and solvable tissue solubilizer was added. 30% hydrogen peroxide was then added during 30 minutes. Afterwards, Ultima gold scintillation cocktail was added to these samples and the resulting mixture was counted for radioactivity, and the percentage accumulated dose per centimeter square values were also calculated. Another technique for the ex vivo testing in oral drug delivery is the everted gut sac technique. The everted gut sac technique was first introduced to study the absorption of compounds along the intestine of rat and golden hamster. However, now it has been explored for the investigation of mechanism and kinetics of drug absorption, prodrug conversion in the GI segments, efflux transport, multi-drug resistance, drug interaction, and also to measure the mucoadhesive properties, transport, and cellular uptake of nanoparticles. In this technique, a section of intestine is quickly removed from the anesthetized animal and flushed with buffer or saline solution and is then everted over a rod or a tube. The serosa becomes the inside of the sac and the mucosa faces the outer buffer solution. Each end of the sac is tied and filled with the oxygenated buffer and placed in the container with the test drug. At the end of incubation, the compounds are analyzed in both the serosal space and the tissue depending on the mechanism of drug absorption. For the compound transported by paracellular route, the test compound will only be in the serosal space and translocation is concentration dependent, while for passively diffusing compounds it is detected in both the serosal space and the tissue, and uptake will be linearly concentration dependent. For compounds having carrier mediated transport, it is detected in both the tissues and serosal space, and uptake may not be concentration dependent owing to the competition by the other substances transported by the carrier. In case of endocytosis, the compound is detected in the tissue and smaller amounts in the serosal space. Several factors may influence the functionality and the outcome of everted sac model. These are animal's age, sex, species, diet, disease or toxicity. The intestinal segment, experimental factors including pH, aeration, temperature, substrate concentration, and other factors such as segment harvesting time and method of killing. The mechanism and preferred site of absorption varies with age. Different species may have different enzymes and transporters. Starvation affects the enzyme activity. Drugs have different sites of absorption. pH affects the drug solubility and ionization. Aeration promotes viability of tissues. Temperature influences viability and mechanism of transport. Quick harvesting of the tissue provides maximum enzyme and transporter activity. Harvesting under anesthesia avoids transporter degradation. The intestinal permeation studies can be carried out in two ways, everted and non-everted gut sac method. The applicability of non-everted rat intestinal sac method has been extensively reported in the literature and has revealed a good relationship between the permeability of model drugs and their corresponding human absorption. In one such report, Tariq et al. Developed epiribicin-loaded biodegradable nanoparticles and evaluated their permeability potential using non-everted gut sac model. According to the procedure, the animals were sacrificed and the ileum was taken out, washed with saline, and one end of the segment was ligated with thread while the other end was mounted on a port of an in-house developed assembly to conduct the study. The sac of each segment was filled with nanoparticles and incubated into pre-warmed and pre-oxygenated thyroid buffer. The samples collected at different time intervals were analyzed using validated analytical method, and P-app was calculated as per the equation, where dq by dt is the rate of drug appearance on the basolateral side, A is the surface area of the intestinal tissue, and C0 is the initial concentration over the apical side. Another technique is everted intestinal rings. In the everted intestinal ring study, the animal's intestine is cut into rings or slices of approximately 30 to 50 milligram and is put into the agitated incubation media, which is properly aerated, similar to the everted sac technique. The model is simple and quick and useful for investigating the uptake into the intestinal cells. Furthermore, the technique offers the advantage of testing several drugs simultaneously and allowing potential screening of the new drug candidate. The method requires short preparation time because rings require no maintenance and a simple apparatus. However, their use is currently limited owing to several disadvantages, which includes drug may absorb onto the surface of the tissue and hence its diffusion may not occur on the mucosal side because the connective tissue and the muscle layers are also exposed in the incubation medium. Additionally, the slices do not maintain integrity for more than 20 to 30 minutes. The human intestinal tissue is now being used to study the intestinal drug permeation. Tissue can be obtained from healthy human sigmoid colon mucosal secretions, including colectomy for colon cancer, angiodysplasia or polyposis, also from biopsies from patients after bariatric surgery. In another techniques, we are going to discuss about the ex vivo studies in ocular drug delivery. The physiological structure, location and sensitivity of the ocular surface predispose it to hazardous exposure from different materials and chemicals, including household, industrial and agricultural products, cosmetics and even certain ocular drugs and pharmaceuticals if incorrectly administered. This can result in damage to the cornea varying from irritation and inflammation, resulting in mild discomfort to ocular tissues and may even lead to irreversible blindness. Thus, all manufactured consumer products and their ingredients need to be tested and their eye irritation potential must be assessed to ensure that the risk associated with the products meets suitable safety criteria and are clearly labeled. The ex vivo test for substances meant to be administered by ocular route are broadly categorized into two categories. First, isolated enucleated organotypic methods. Second, non-organotypic methods. Now the first is isolated enucleated organotypic methods. Organotypic enucleated models are borderline between the in vivo and the in vitro systems, requiring lesser ethical connotation with reduced costs. These are the isolated systems which maintain short-term normal physiological and biochemical functions of the enucleated eye or cornea and more closely represents accidental exposure. According to the protocol established, the quantitative assessment of changes to the isolated cornea in response to the test material is made using opacitometric and spectroscopic methods followed by histological analysis. Corneal opacity serves as an indicator of protein denaturation, swelling, vacuolation and damage to the epithelium and corneal stroma. Permeability is measured with the assessment of fluorescein retention or leakage of the cornea. The histological analysis is included as an additional endpoint, as the iris and the conjunctiva are also involved in the ocular irritation and swelling. The eye irritation is based on the depth of initial injury as shown in the figure and correlates with the extent of cell death and ultimately the outcome of the irritant on an eye. Generally, the superficial epithelium is damaged by the slight irritants. Mild irritants penetrate further and leads to the damage of stroma, while the severe irritants penetrate through the cornea, damaging the endothelium. Ocular organotypic models or enucleated eye test were first carried out using isolated rabbit eyes from animals used for other research purposes or those sacrificed commercially as a food source. Slaughterhouse waste is also used as an alternative tissue source for EETs. Chicken enucleated eye test, also known as the isolated chicken eye test, are widely accepted to be a reliable and accurate slaughterhouse tissue for assessing the test material's eye irritation potential. The ICE testing protocol is based upon the IRE model. The eyes are isolated from an intact chicken head and processed 2-hour postmortem. The enucleated eye is positioned in a clamp with the cornea positioned vertically and transferred to a superfusion apparatus for determining the damage. Once approved, the eyes are equilibrated for up to 1 hour and baseline thickness and opacity measurements are recorded. The eye is then positioned horizontally and the test substance is applied for 10 seconds. The cornea is further rinsed with hypertonic saline before being returned to the superfusion chamber for analysis. Toxic effects are recorded by measuring changes in opacity, fluorescein retention, tissue thickness and a macroscopic evaluation of the changes to the surface of tissue. Another ex vivo assay is bovine cornea opacity permeability assay. BCOP testing has been found to be scientifically valid and suitable in identifying both the substances that induce serious damage and those that are classified as non-irritants, and has received acceptance with the regulatory bodies such as ICCVAM in conjunction with the European Union Reference Laboratory for Alternatives to Animal Testing and the Japanese Center for the Validation of Alternative Methods. In the BCOP assay, the O-rings are mounted over a chamber between which the intact corneas of the healthy animals are placed. An anterior chamber is positioned above the cornea, both of which are clamped together. Each chamber has its own dosing hole that allows both the epithelium and the endothelium to be treated independently. Opacity is measured using an OPKIT opacitometer. The porcine cornea opacity permeability assay using porcine corneas are advantageous compared to bovine corneas due to lesser concerns of encephalopathy disease, closer resemblance to the human corneas with regard to the structure and thickness, and their regular use in the ophthalmic research. The preliminary studies with the PCOP reveal that it can be used to accurately predict eye irritation for liquid and water soluble substances. However, it is not yet adopted by the regulatory bodies that seems to favor BCOP. The EETs have demonstrated promising potential with regard to the testing of ocular substances. However, they suffer with several limitations accounted as interspecies differences regarding anatomy and physiology producing discrepancies in permeation studies and toxicity tests. They do not consider conjunctival and irradial issues, inflammatory response elements and corneal recovery or reversibility of lesions, cannot predict systemic effects of substances such as lethality of certain pesticides. They can only be used for relatively short-term assessment periods and so are not suitable for testing substances that produce effects over extended time frames. Another category is non-ocular organotypic models. The non-ocular organotypic models includes two types of ex vivo studies, the CAM assay and the SMI assay. The chorioallantoic membrane vascular assay, also known as hen's egg test or simply CAM assay, is used to provide qualitative information on the potential effects occurring in the conjunctiva following exposure to a substance, whilst evaluation of coagulation can be used to reflect potential corneal damage. CAM is the vascularized respiratory membrane found within the membrane of a fertilized chicken egg with a vasculature and inflammatory process similar to the conjunctival tissue of rabbit's eye. The general protocol described in the figure involves exposing the cam and applying the test material to the surface, followed by rinsing and observing changes to the membrane morphology, which are assessed and scored. The protocol can be varied depending upon the incubation times, relative humidity, number of replicates, breed of hen, egg rotation, method of opening the eggshell, use of positive negative controls, and exposure time. The time whereby a substance causes hemorrhage, vasoconstriction or coagulation is measured, scored and categorized. The CAM assay has yet to receive international regulatory acceptance. However, ICC VAM recommends that the test is used for non-regulatory validation or optimization studies. The other non-ocular organotypic model for studying ocular irritation is slug mucosal irritation assay, called as SMI assay. It uses the terrestrial slug which produce mucus and lose body weight when placed upon the irritating surfaces. The slugs also release additional proteins and enzymes from its mucosal surface when tissue damage occurs. The evaluation criteria lies on the extent of mucus production, as the mild irritants cause an increase in mucus production, whereas severe irritants result in the tissue damage and protein release in addition to the increased mucus production. However, the SMI assay failed to pass a formal validation study, so it is currently only used as a pre-screen for simple toxicological endpoints. Now we are going to discuss about the ex-vivo models used in transdermal drug delivery. Dermal and transdermal drug delivery have been investigated as an attractive alternative to conventional oral therapy to avoid problems such as first-pass metabolism, side effects and poor patient compliance, and also to the parenteral therapy, which is painful, generate dangerous medical waste and pose the risk of disease transmission by needle reuse. The transdermal systems are non-invasive, can be self-administered, provide release for long period of time and are generally inexpensive. The assessment of percutaneous absorption of molecules is a very important step in the evaluation of any dermal or transdermal drug delivery system. Ex-vivo methods which predict human dermal penetration are available, offering advantages over the whole animal or human volunteer experiments. The advantages may include saving in time and cost, better reproducibility of the results, less restricted parameter variations. They can be used equally well with skin from the humans and other species. Several replicate measurements can be made from the same or the number of different subjects. The non-radio-labeled test substances which are extensively metabolized can be studied. These are designed to measure the penetration of chemicals into and subsequent permeation across the skin into a fluid reservoir. A non-viable skin can be used to measure penetration and permeation only, and a fresh metabolically active skin can be utilized to simultaneously measure permeation and skin metabolism. Special consideration should be given to type of cell system, skin source, viability and composition of the receptor fluid. A possible limitation of these systems is that the skin conditions of the peripheral blood flow may not be fully reproduced. According to the protocol, the test substance in an appropriate formulation is applied to the skin surface, which is mounted as a barrier between the donor compartment and the receptor compartment of a diffusion cell. Diffusion cells may be of static or flow-through cells. Static diffusion cells sample this chamber and replace with the new perfusate at each time point. Flow-through cell use a pump to pass perfusate through the receptor chamber and collect flux by repeatedly collecting perfusate. The static diffusion cells can be further categorized on the basis of skin orientation, with the membrane being placed horizontally or vertically. Majorly, the skin absorption studies are conducted using horizontal cells with the skin surface open to the air, while the vertical cells are used for the evaluation of drug delivery systems such as sonophoresis, iontophoresis or electroporation, and requires immersion of both the surfaces of skin preparation that may cause excessive hydration and skin damage. The receptor fluid should be temperature controlled, have adequate capacity to solubilize the test substance, be maintained in contact with the underside of the skin from the time of application until the end of the collection of receptor fluid, and well stirred throughout the study. Saline or buffered saline solutions are commonly used for hydrophilic compounds, while serum albumin or appropriate solubilizers or emulsifiers having non-interference with the membrane integrity are selected for the lipophilic molecules. The receptor fluid should have a physiological pH. The amount of penetrated substance in the receptor fluid should not exceed 10% of its saturation level at any time, so as to minimize interference with the free diffusion process that could produce an underestimation of the dermal absorption. The skin sample selected should not have any physical damage and allowed to equilibrate with the receptor fluid for typically 10 to 30 minutes before dose application. The barrier integrity of the skin should be checked by physical methods such as transepidermal water loss or transcutaneous electrical resistance. There are two main types of dosing regimes which are generally used in skin absorption studies. First is the infinite dose, in which the typical dose of 100 microliter per centimeter square or greater than 10 milligram per centimeter square is used, and finite dose, in which up to 10 milligram per centimeter square or 10 microliter per centimeter square is used. In the infinite dose experiments, the concentration of the chemical present on the skin surface effectively remains constant. This is not a realistic scenario for most skin exposure to the chemicals, but this largely artificial condition is frequently used to evaluate the important kinetic parameters such as flux, permeability coefficient and diffusion coefficient. A finite dose condition more closely represents the actual dermal exposure, where a discrete quantity of substance is applied to the skin surface and is gradually depleted with time. For semi-solid preparations including creams, paste or other viscous test preparation, a spreader may be required to evenly distribute over the application site. To mimic the normal exposure conditions, the skin surface may be unoccluded, semi-occluded or occluded. The unoccluded conditions allows any volatile component of the test preparation to evaporate and the normal drying process to occur. A second effect of occlusion is to enhance the skin hydration by preventing the normal loss of water from the surface of skin. Thus, the unoccluded or semi-occluded conditions can avoid skin integrity damage that can be caused by the excessive hydration which may occur with occlusion. Duration of the study over 24 hours may result in membrane deterioration and requires careful control of membrane integrity. The frequency of sampling should be chosen adequately to allow the determination of extent or rate of absorption and the absorption profile. At least 6 post-application time points should be included to estimate the adequate absorption kinetics. The terminal procedures for the study are slightly different for the infinite and finite dosing experiments. After finite dosing, the mean maximum amount of dermally absorbed material is determined, requiring complete recovery of the test substance. According to OECD test guidelines, the test substance remaining in the skin should be considered as absorbed unless it can be demonstrated that the absorption can be determined from the receptor fluid alone. Also, when the test substance remain in the skin at the end of the study, it may need to be included in the total amount absorbed. For infinite dose application, the steady state flux is determined and permeability coefficient is calculated. Now we are going to discuss the different ex-vivo models used in the nasal drug delivery. Drug delivery using nasal route is gaining wide acceptance within the pharmaceutical industry owing to the potential advantages associated with this system. These include: first, availability of relatively large surface area for drug absorption; second, rapid drug absorption and onset of therapeutic action due to the presence of thin, porous and highly vascularized epithelium with enhanced blood flow; third, direct delivery of absorbed drugs into systemic circulation; fourth, efficient bypass of the first pass effect of liver and GIT; fifth, greater patient compliance; sixth, possibility of pulsatile and controlled drug release from the system. Determination of drug permeability across a specified mucosal epithelium and screening of permeation enhancement and toxicity plays a very crucial role in selection of the compounds early in research and development. Therefore, the use of in situ nasal perfusion excised human or animal tissues and cultured cells as drug screening models for determination of drug permeation, metabolism and toxicity is growing in popularity. The set of information provided with the excised tissues or cultured nasal cells are: solute transport across the nasal epithelium, effect of enhancers and their concentration on the flux, histological examination for determining the extent of tissue damage, respective mechanisms involved, and the effect on the ciliary beat frequency. A Singh chamber technique similar to that mentioned above is used. The tissue samples from the rabbits, pigs, cows and sheep are most commonly employed. The chicken embryo trachea has been also investigated for the determination of mucociliary clearance. MUC5AC and MUC5B imparts gel forming and adhesive properties to airway mucus. Alterations in secretion rate and MUC5AC to MUC5B ratio correlates with the impaired mucociliary clearance, resulting in several pathophysiological conditions of the respiratory system. This model allows exploring mucociliary clearance and mucus particle interactions based on the functional interactions of cilia and mucus. It has been established that the embryonic chicken trachea is a valid substitute for the human material in studying ciliary beat frequency and ciliary toxicity. Chicken eggs of SPF quality are incubated in a breeding chamber for 19 to 20 days. After dissection, the trachea is placed on gauze soaked with Lochringer solution. The tracheal tube is cut oblong, resulting in 2 tracheal half pipes. Particle deposition on the half pipes is realized using a dry powder insufflator. Following particle deposition, the tracheal tissue was transferred to the incubation chamber with standardized condition and subsequently placed under the microscope. Particle transport on the trachea is recorded. The tracheal tissue is also subjected to histopathological analysis and determination of clearance velocity. Although the ex vivo studies are finding wide utility in the screening and evaluation of therapeutic molecule, they have several limitations which should be considered while extrapolating the results for the in vivo studies. The animal species differ in the thickness of the nasal epithelia tissues, which is further complicated by the amount of connected tissue left on the serosal side of the epithelium after tissue preparation. The current lack of standardization of the experimental conditions with respect to the temperature, gas perfusion, tissue incubation medium and the stirring conditions further complicates the result for the in vivo extrapolation. The drug migration into the perspex body of the Asing chambers should also be taken into account. Use of liquid mucosa interface compared to the air mucosa interface can further contradict the in vivo situation. Additionally, the absence of mucus layer and ciliary clearance mechanisms complicates the situation. The species also differ in the type of metabolic enzymes and their metabolic capacity present in the nasal epithelium. The safety of the excised tissue should also be taken into consideration. Example, tissues from the bovine origin can have a problem of bovine spongiform encephalopathy. The utility of ex vivo studies in current research can be exemplified with the study of Abdul Bari et al. They formulated olanzapine loaded micellar nanoparticles for nose to brain targeting and conducted histopathological studies on the isolated sheep nasal mucosa to determine the possible local toxic effects following nasal instillation. According to the procedure, the sheep nasal cavity mucosa was obtained from the slaughterhouse. Within 15 minutes of the animal sacrifice, the nasal cavity was exposed by a longitudinal incision through the lateral wall of the nose, avoiding damage of the septum. The mucosa was carefully removed and immediately placed in a 900 ml ice cold Ringer solution. 2 segments carefully separated from the interior and the posterior regions of the mucosa was sectioned into 3 pieces, and each piece was treated with olanzapine loaded micelles, phosphate buffer saline pH 6.4 which served as the negative control and isopropyl alcohol which served as the positive control. 2 hours post treatment, they were washed with distilled water and preserved in 10% formalin solution for 48 hours and subjected to histopathological examination, where the samples were dehydrated by treatment with serial dilutions of methyl and ethyl alcohol and absolute ethyl alcohol. Specimens were cleared in xylene embedded in paraffin in hot air oven with temperature adjusted at 56 degrees centigrade, and the samples were kept for 24 hours. Paraffin beeswax tissue blocks were sectioned by a sledge microtome, collected, deparaffinized, stained by hematoxylin and eosin, and examined under the light microscope. The next are the ex vivo models for buccal drug delivery. Buccal drug delivery across the mucosal lining of the cheeks offers several advantages over the other routes of drug administration. These include avoidance of pre-systemic drug elimination within the GIT, and or during the hepatic first pass metabolism, averting the potential variability in drug absorption, which is mediated by the gastric emptying rate or the presence of food in the upper regions of GIT. The third is the substantial resistance to irritation or damage due to rich blood supply, making it more permeable. However, despite the potential advantages, the buccal administration has been utilized for relatively few pharmaceutical products so far. This may be attributed due to costly preclinical development, which includes laborious pharmacokinetic evaluation in animal models. Thus, suitable in vitro methodology can greatly aid in the early development by subjecting greater number of compounds or formulations for the evaluation of their potential for buccal absorption. The most widely used methodology for assessing drug permeation across buccal mucosa is by using isolated buccal tissue mounted in diffusion chambers. The diffusion cells helps to determine the actual amount of drug that diffuses across the mucosal barrier and the rate of drug diffusion. Several types of diffusion cells are available, among which France type diffusion chamber are the most widely utilized for the determination of buccal permeation. The construction and working of the France diffusion cells are the same as discussed previously. Selection of skin from animal species is of crucial importance for the permeation studies. Rabbit has been widely used in buccal absorption studies as it is the only laboratory animal with a non-keratinized surface layer of the buccal mucosa similar to that in the human tissue. However, non-keratinized area of the buccal tissue of rabbits has a sudden transition to keratinized regions, which limit the amount of suitable tissue available and hence makes isolation of the desired region unreliable. Therefore, buccal mucosa of monkeys, dogs and pigs has been extensively used for in vitro drug permeability studies. The porcine buccal tissue is the most frequently utilized model owing to the relative similarity to human tissue and a low cost associated with the tissue acquisition. The physicochemical properties such as lipophilicity, molecular weight and degree of ionization at physiological pH of the drug molecule influences drug permeation across the buccal tissue. Therefore, ex-vivo model for estimation of drug permeation across buccal mucosa are ascertained with several benefits, which may include assessment of the buccal delivery at the intended application site, where unlike the in vivo situation, there is no compound loss due to swallowing. The second benefit is a wide range of excipient testing attributed to the tissue resistance to irritation and loss of integrity, and the possibility to obtain in vitro animal buccal tissue data that can be correlated to the in vivo animal results prior to human testing. So students, we have studied about the different techniques used in the ex vivo studies for the products meant to be delivered by different routes. They can serve as a measure of high throughput screening of the therapeutic molecules in the drug development process. This is all in this module. Thank you.